All right, everyone, now we're going to take a look at chapter 31. In this case, we're going to look at plant responses to signaling. So there are certain stimulus that a plant receives and how it responds. It's not like our animal hormones, but they do have their own signaling molecules, and it's important to understand how they function as well. So one of the things that was done with plants, just as an intro experiment, and I'm talking about it because this is kind of the order that the book talks about it, but is looking at what we call coleoptiles. And these are kind of the shoots that come out of seeds when they're about ready to grow. And what they found was that when they shined light on these particular shoots, that they actually grew towards the light. And what happens is the shaded side, or the side that doesn't have light, actually elongates compared to the side that doesn't, or that does get light. And so what happens is the light side has shorter cells, the dark side has longer cells, and when that happens, it will then cause a bending towards the light. And there were various other things that would happen to other experiments. So you could try this with a permeable seed and an impermeable seed, and it just kind of showed you that it did need to have light making it into the cells to allow photosynthesis to happen, and that these specific cells did need to be exposed to the light, because if those specific cells in the tip were not there or prevented from having any kind of light, then curvature could not take place. So the conclusion was that the tip of these shoots are able to sense light, and that some kind of signal that is sent from the tip to the elongation region causes that change in direction. The specific chemical that was found to actually stimulate this elongation was called auxin. And as I mentioned, the cells on the darker side have a tendency to elongate faster than the ones on the brighter side. And auxin is the chemical that is released and causes the elongation in those darker cells. And the graphic isn't super great here, but the idea is that you took the tip off, in this case, and placed it on an agar block. And what happens is the growth promoting chemical, in this case auxin, went into the little agar block. And they wanted to see if that chemical could make its way and cause the plants to change. So if you just straight up cut the top off, that's the control, right? There's no more auxin anymore. That's in the tip. So therefore, it can't actually cause any movement. And if you never introduced the tip, then no chemical can make its way into the agar block. Therefore, there's no effect. However, in the middle, what they found was that if you specifically have it equal on both sides, there's no real shift, because you have auxin on both sides of the plant, and therefore it's causing equal elongation on both sides. However, if you actually put like a half a block, so you expose auxin to one singular side of the plant, it was enough to actually cause curvature. So that shows that the auxin chemical is acting in those elongation cells on the dark side of the plant. Plants have a number of hormones that they use to do different things. Auxin was one that we just talked about, and it stimulates cell elongation. So it causes different tropisms, which we'll talk about in a minute, but it has to do with movement. So phototropism is the movement of a plant towards light. Gravitropism is the movement of a plant due to gravity. And a lot of times, high concentrations of auxin are found in herbicides because they can hurt plants as well in super high concentrations. There are also other categories like cytokinins, in which case they cause cell division, and cytokinesis, which is why they are caused that, so it increases that. Gibberellins are growth hormones. So they trigger fruit growth, is kind of the main thing that they're used for. They're used by farmers a lot of times to increase fruit size. Abscisic acid slows growth. So this is oftentimes to prevent or promote dormancy. So when you don't want something to grow, it also triggers the closing of stomata, which open up on the underside of plants, if we remember, to allow gas exchange for photosynthesis, as well as act as a source of water loss. And then the last one is ethylene, which is a chemical that promotes fruit ripening and acts kind of in a positive feedback capacity. So if the presence is there, it causes fruit to ripen, which causes more fruit to ripen, which causes more fruit to ripen. And it's involved in apoptosis. So this has to do with the ripening of fruits, as well as the death of plants come the end of a season if they are annuals, meaning they come back every year, or you plant them every year. My mistake. So here's an example of gibberellins on fruit growth. So they do trigger stem elongation. So if in their presence they trigger these shoots to come out. On the right, you can see on the left would be your typical organic, no hormone grapes. On the right, you can see those ones have been treated with gibberellins. So a lot of farmers, as I mentioned, treat fruit with the gibberellin hormone, and then that increases fruit size. Ethylene gas is another example for fruit ripening. So this is actually released when you put, or one of the reasons why when you put fruit in a paper bag, it triggers ripening because it kind of blocks that gas in, and because it's a positive feedback, it'll help promote that process. And it's interesting too because transporters of bananas, if they are unripe, actually just keep canisters of this particular gas and release it in the truck. And then that will allow the fruit to ripen faster, so it's ready to go when it makes it to market. And on the bottom, these are tomatoes. You can see if they've been treated or untreated, it helps them ripen faster for when they need. So, as I told you before, tropisms have to do with growth. It's not a fast process, but it does have a growth response. Phototropism, as I mentioned, is due to light, and this is in response to the hormone auxin, which stimulates the elongation on the dark side of plants. And it can be positive, meaning that it grows toward the light, like shoots that are going to be doing photosynthesis. But interestingly, there's also negative phototropism, meaning that it wants to go away from the light. So this is what roots do when they're going down into the ground. They're essentially going away from light. Auxin can also control gravitropism, which is due to gravity. So it's the elongation of cells and kind of the opposite form of gravity, so that a lot of times plants end up getting weighed down and grow down in the path of gravity. In a minute, I'll show you what Thigmotropism is, but it's a response in relation to the touch stimulus. So some plants actually move in response to touch. There's also turgor movement, which has a specific rapid response that can be reversible. So the Venus flytrap is kind of the quick, easy go-to for that type of thing. If you can imagine, like, the fly lands on a Venus flytrap and then it snaps shut, and then eventually it'll open itself again. So that's the quick process that a plant could do in response to the stimulus of a fly landing on the leaf. Here's an example of what gravitropism might look like. And you can see kind of before and after on the bottom, the root of maize, or corn in this case, is bending due to gravity. And those cap cells have specific hormones that are settling onto the lowest sides of the root cap. And because of it, it kind of drags this thing down and weighs it down so it shifts towards the light. Again, this is controlled by auxin. The touch response, thigmotropism. The mimosa plant is one where it causes actual action potentials. So this is a signaling that's not G-coupled, but is in relation to action potentials or ligand gated ion channels and can actually cause the stimulation to move the plant in a different direction. So you can kind of see what it looks like unstimulated versus stimulated. Okay. In response to light, plants can detect direction, intensity, and wavelength to light. There exist special things called phytochromes that are light receptors inside plants that absorb mostly red light. So we oftentimes think of pigments to absorb light and fuel photosynthesis, but there are also some things called phytochromes that are light receptors, and they absorb light that can regulate seed germination as well as shade avoidance. So if you expose a seed to light, or you expose, in this case, a potato to light, eventually it will sprout. So essentially you're germinating the plant to grow, because the light is an indicator for, like, let's go photosynthesis. After a weak exposure, you can see what the plant might look like on the right. So these things kind of help to regulate the initial signaling to get plants growing. What's interesting is that plants have biological clocks. So we as humans have biological clocks as well. We have circadian rhythms, and that's why we typically exist on a 24-hour schedule, and we persist and oftentimes do that without environmental cues. So if we had no concept of time, our bodies would kind of naturally get sleepy and awake at the same time every day. But what's interesting is plants can use that light sensing system, the phytochromes, along with the biological clock idea to figure out what time of year it is. It can get a sense of what time of year it is based on the hours of daylight that exist outside. And certain plants prefer to bloom and or grow in different times of year. So based on the phytochrome sensing how much light is outside in a given day, it can say, okay, yes, this is my prime time, let's bloom, or no, I need to wait longer, or my time is done. And this leads to something called photoperiodism, where plants are blooming at different times of year because of the natural length of the day. So there are short day plants, which means that they are growing mostly when the nights are long. So mums and poinsettias are examples. These are the ones that will grow for us in the US in fall and winter time, because that's when the nights are longer and the days are short. Other plants like spinach, lettuce, radishes, things like that, grow in the peak of summer. So they are the long day plants. They like it best when the nights are short. And then there's ones that are day neutral, that really don't care at all. They don't need a necessary amount of light. But the idea here is that the night length is super critical. And here's kind of an example experiment showing how that is. So for a short day plant, for example, they are the ones that like the short day. So this middle bar here is showing you how the plant can grow. Short day, long night, perfect. If the day is longer than 12 hours, that's where this dotted line is, it will not grow, too long. If the day is short, but then somehow they get a flash of light during that critical nighttime period, it won't grow. So that highlights the fact that the high level and high amount of darkness is actually important. Whereas a long day plant, if the day is longer than 12 hours, the plant is happy. If it's less than 12 hours, it is not happy. If it's less than 12 hours but somehow gets a flash of light, it will be happy. So if you interrupt the really long nighttime period, that's enough to trigger the long day plant to grow. This slide I'm just going to kind of gloss over because you can mess with things in the nighttime, but to be fair, you don't really need to go into that much detail. And the tail end of this PowerPoint is really plant response to stress. We have kind of talked about this previously, but just kind of thinking about what plants do in response to various stimulus. So these are stress stimulus to drought conditions. We kind of talked about how there's adaptations to close the stomata. They can slow their growth. They can deepen their roots. If there's too much water, because you can overwater a plant to death, they might do things like release ethylene. They will kill off some of their roots, and so on and so forth, to try to adapt as much as they can to the situation. If there's too much salt or heat or cold, they have adaptations for that as well. And you can kind of read those at your own pace. Some of them are quick, others are more long-lasting, like specific proteins or specific plasma membrane compositions. Plants have responses to herbivores that oftentimes like to eat them. So they have physical barriers like thorns, they have chemical barriers like toxins that they release, and so on. And they also have lines of defense against the stimulus of pathogens. And we're going to end up talking about the immune system a little later on. So just to give you a sense of how they react to their environment. And that's it.